Every second, somewhere inside your body, a skin cell is dividing. Its nucleus, a sphere smaller than a dust mote, contains a molecule just over 2 meters long, folded and coiled into a space 10,000 times smaller than its stretched length. That molecule is being read right now, copied, checked for damage, repaired where necessary, and the same process is happening simultaneously in roughly 37 trillion other cells, scattered through your bones, your blood, your brain. If you could somehow gather all that DNA from all those cells and lay it end to end in a single thread, it would stretch for 74 billion kilometers. That is far enough to travel from Earth to the Sun and back not once, not 10 times, but roughly 245 times. One human body contains a molecular archive that spans the inner solar system. And yet, when that skin cell finishes copying its entire 2-meter blueprint, all 6 billion chemical letters, it will introduce on average fewer than 6 errors. The machinery responsible for this feat operates with an effective error rate of roughly 1 mistake per billion characters replicated. No human technology comes close. The Mars rovers, marvels of engineering, carry error correction systems for their onboard memory that would be considered robust if they achieved 1 error per 10 million bits. DNA replication is 100 times more accurate, performed by molecular machines no wider than a few nanometers, working in the warm, chaotic environment of living tissue. This is not a careful process happening in a clean room under controlled conditions. This is happening now in you while your cells are being bombarded by radiation, by metabolic byproducts, by the simple thermal chaos of being alive. Each cell's DNA suffers more than 10,000 separate injuries every single day, breaks, corruptions, chemical modifications that would render the message unreadable if left unrepaired. And yet the genome endures. It is read correctly. It is copied faithfully. Generation after generation, the instructions persist. The question that will carry us through the next 2 hours is not whether this system is remarkable. The question is how. How does a molecule that long stay both compact and reliable? How is it copied with such fidelity? And when it is damaged, as it constantly is, how is it repaired before the error becomes permanent? If this is the kind of thing that pulls you in, that makes you want to understand the machinery beneath the surface of life, you're in the right place. Settle in. Let's begin. Stand for a moment inside a single skin cell on the back of your hand. You're in the cytoplasm, the thick molecular broth that fills the space between the cell's outer membrane and its nucleus. Around you, ribosomes are translating messenger RNA into proteins. Mitochondria are splitting glucose and oxygen into the chemical currency the cell runs on. But the center of this space is dominated by the nucleus, a membrane-bound vault roughly 6 micrometers across. It looks, under the right stains, like a dark sphere suspended in the cell's interior. That darkness is not emptiness. It is density. It is information. Inside that nucleus, coiled and folded with obsessive geometric efficiency, is the cell's entire operating manual. 3 billion base pairs arranged in a double helix, wrapped around protein spools, bundled into loops, and compacted until 2 meters of linear molecule occupies a volume smaller than a red blood cell. The nucleus is not a static library. It is alive with motion. Proteins are sliding along the DNA, reading genes. Enzymes are peeling the 2 strands apart to copy them. Repair machinery is scanning for damage and excising corrupted sections, patching them with fresh nucleotides synthesized moments earlier. The genome is not a book on a shelf. It is a script being performed in real time, and the performance never stops. Now pull back. That one skin cell is not unique. The same 2-meter thread, the same 6 billion letters, exist in nearly every other cell in your body, in your liver, in the lining of your gut, in the neurons that are right now processing these words. 37 trillion copies of the same message, each one coiled into its own microscopic nucleus, each one being read and maintained independently. The total length of that molecular thread, if you could extract it all and lay it in a straight line, would reach halfway to the orbit of Jupiter. One human being is, in molecular terms, a solar system scale archive, and this archive is not passive. It is under attack. Ultraviolet light from the sun breaks chemical bonds in the DNA of your skin cells. Free radicals, byproducts of the cellular respiration happening in your mitochondria, collide with the DNA backbone and modify its structure. Even the thermal energy of your body, the ordinary warmth of being alive, is enough to occasionally snap a chemical bond or knock a molecular group off a nucleotide base. The genome is not sitting safely behind glass. It is out in the weather, taking damage every moment of every day. And yet it persists. It is copied with such fidelity that the instructions for building a human being can pass from generation to generation across hundreds of millions of years of evolutionary time, accumulating only the changes that matter, the ones that are not immediately fatal. The machinery that makes this possible is not one enzyme, not one system. It is an overlapping network of proofreading steps, backup mechanisms, and repair pathways, each one capable of recognizing a different kind of damage and correcting it before the error is locked into the next generation of cells. The effective error rate, 1 mistake per billion bases copied, is not the natural rate. It is the rate after 3 layers of correction have been applied. The molecular machine that copies DNA, DNA polymerase, works at an intrinsic error rate of roughly 1 mistake per 200,000 nucleotides. Still impressive by human engineering standards, but not enough, not nearly enough. So the polymerase carries a proofreading function that catches most of its own mistakes immediately, dropping the error rate by another factor of 100. And if an error still slips through, a separate system called mismatch repair patrols the freshly copied DNA, finds the errors the polymerase missed, cuts them out, and fills the gap with the correct sequence. That is how you get to 1 error per billion. That is how the genome of a skin cell, after copying 6 billion letters, emerges with fewer than 6 mistakes. And that error rate has to be that low. Because you are not one cell. You are 37 trillion cells, and they are dividing constantly. Skin cells every few weeks, gut lining every few days. Over a human lifetime, the genome is copied so many times that even a 1 in a billion error rate means trillions of mutations across the body. Most are harmless, caught in cells that die without consequence. Some are not. The difference between a mutation that does nothing and a mutation that begins a tumor is often a single letter in a single gene. The machinery that maintains the genome is not just impressive, it is load-bearing. It is the reason multicellular life at our scale is possible at all. But before any of this machinery could be understood, before anyone could ask how the genome was copied or repaired, someone had to find it. And for most of human history, no one knew DNA existed. The year is 1869. The place is Tübingen, a medieval university town in southwestern Germany, built on a hill above the Neckar River. The old Hohen-Tübingen castle overlooks the town, its stone walls dating back centuries. Inside one of the university's laboratories, a young Swiss physician named Friedrich Miescher is working with pus. Miescher is 24 years old. He has just begun a research position under Felix Hoppe-Seyler, a prominent biochemist interested in the chemistry of living tissue. Miescher's interest is more specific. He wants to understand the chemical composition of cells, and in particular, the contents of the cell nucleus. To do this, he needs a large supply of cells with prominent nuclei, and he has found an efficient if unpleasant source. He collects surgical bandages from a nearby clinic, bandages soaked with pus from patients recovering from surgery. Pus, it turns out, is rich in white blood cells, and white blood cells have large accessible nuclei. Miescher's process is painstaking. He washes the bandages, isolates the cells, treats them with a weak solution of sodium sulfate to break down the cell membranes, and then adds dilute acid to dissolve away the proteins. What remains is a greyish precipitate that does not behave like any substance he has seen before. It is rich in phosphorus, which is unusual. Proteins typically contain carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur, but not much phosphorus. This substance also lacks sulfur, which is a defining feature of many proteins. When Miescher tests it further, he finds it is not a protein at all. It is something else, something new. He calls it nuclein, because it comes from the cell nucleus. He writes up his findings in 1869, but publication is delayed. Hoppe-Seyler, cautious and methodical, insists on repeating the experiments himself to verify the result. It takes 2 years. Miescher's paper finally appears in 1871, and the substance he isolated, nuclein, enters the scientific record. But no one knows what it does. It is simply a phosphorus-rich material found in cell nuclei, chemically distinct from protein. It will be another 75 years before anyone proves it carries the instructions for life. In the years after Miescher's discovery, other researchers begin to notice that the nucleus is not just chemically interesting, it is structurally active. In the late 1870s, a German anatomist named Walter Flemming working at the University of Kiel, begins staining cells with aniline dyes. These dyes, derived from coal tar, have the useful property of binding to certain cellular structures and making them visible under a microscope. Fleming is particularly interested in what happens inside the nucleus during cell division. What he sees is striking. Just before a cell divides, thread-like structures appear inside the nucleus. They condense, align themselves along the cell's midline, and then split, with half moving to each daughter cell. Fleming names these structures chromatin, from the Greek word for colour, because they absorb the dye so readily. He meticulously documents the entire sequence of events and publishes his findings in 1882 in a book titled Zellsubstanz, Kern- und Zellteilung, cell substance, nucleus, and cell division. He calls the process mitosis, from the Greek for thread. Fleming establishes a principle: all cell nuclei arise from a predecessor nucleus. It is an echo of Rudolf Virchow's earlier dictum that every cell comes from a cell, now applied to the nucleus itself. What Fleming does not know, what no one yet knows, is that those threads he is watching are made of Misch's nuclein, and that they carry something far more important than structure. They carry information. At the same time, Fleming is watching chromosomes divide in Germany. A set of experiments conducted 2 decades earlier is sitting, unread, in an obscure journal. Between 1856 and 1863, an Augustinian friar named Gregor Mendel, working in the garden of a monastery in Brunn, now Brno in the Czech Republic, bred and crossbred roughly 29,000 pea plants. He tracked 7 traits: seed shape, seed color, flower color, pod shape, pod color, flower position, and plant height. And he discovered something extraordinary. The traits did not blend. When Mendel crossed a tall plant with a short plant, the offspring were not medium-sized. They were tall. And when he crossed those tall offspring with each other, 3/4 were tall and 1/4 were short. The short trait had not disappeared. It had been hidden, masked by the presence of the tall trait, and it reappeared intact in the next generation. Mendel called the masking trait dominant and the hidden trait recessive. He worked out mathematical ratios. He inferred that each parent contributed 1 factor for each trait, and that these factors segregated independently during reproduction. He presented his findings to the Natural History Society of Brunn in February and March of 1865. The paper was published the following year in the Society's proceedings, and then, for 35 years, almost no one paid attention. The work was too mathematical for most biologists, too abstract, and it appeared in a regional journal that few scientists outside Central Europe ever read. Mendel himself moved on to other duties. He was elected abbot of his monastery in 1868 and spent the rest of his life managing the institution's finances and disputes with the government over taxes. He died in 1884, his work on inheritance largely forgotten. But in 1900, 3 botanists working independently, Hugo de Vries in the Netherlands, Karl Correns in Germany, and Erich von Tschermak in Austria, each rediscovered Mendel's laws while conducting their own breeding experiments. Each searched the scientific literature and found Mendel's 1866 paper, and suddenly, 35 years too late, Mendel's factors became the foundation of a new science. They were renamed genes, and biologists realized that Fleming's chromosomes, those thread-like structures visible under the microscope during cell division, were the physical carriers of Mendel's inherited factors. Biology now had a ledger. The traits that passed from parent to offspring were not vague vital essences. They were discrete units, carried on physical structures inside the cell nucleus. Misch's nuclein, Fleming's chromatin, and Mendel's factors were all describing different aspects of the same system, but the connection between them remained indirect. No one knew what genes were made of, no one knew how they were copied, and no one knew how they produced the traits they controlled. The physical ledger had been found, but its language was still unreadable. That language begins to come into focus in the early 20th century, but only slowly. Biochemists work out that nuclein, now called nucleic acid, comes in 2 forms, one found mainly in the nucleus, which they name deoxyribonucleic acid, and one found mainly in the cytoplasm, ribonucleic acid. DNA and RNA. Chemical analysis reveals that DNA is built from 4 types of smaller molecules called nucleotides, each one containing a sugar, a phosphate group, and one of 4 nitrogen-containing bases: adenine, thymine, guanine, or cytosine. A, T, G, and C. 4 letters. But for decades, Most scientists assume DNA is too simple to carry genetic information. The prevailing hypothesis, known as the tetranucleotide hypothesis, holds that DNA is just a monotonous repeating structure, perhaps some kind of scaffolding that holds the real genetic material, proteins. Proteins, after all, are built from 20 different amino acids. That gives them the complexity and variability that genes would seem to require. DNA, with its 4-letter alphabet, seems too limited, too uniform. And so the majority of researchers in the 1930s and early 1940s believe that genes are made of protein, and that DNA is merely structural, a molecular skeleton with no informational content. It takes an experiment conducted in the middle of a world war to overturn that assumption. The Rockefeller Institute for Medical Research in New York is one of the premier centers for biological research in the world. In the early 1940s, a team led by a quiet, methodical bacteriologist named Oswald Avery is working on a question that has puzzled microbiologists for nearly 2 decades. In 1928, a British researcher named Frederick Griffith discovered that a non-virulent strain of Streptococcus pneumonia, a bacterium that causes pneumonia, could be transformed into a virulent strain simply by exposing it to heat-killed virulent bacteria. Something in the dead virulent cells was being taken up by the living non-virulent cells, and that something was changing them permanently. Griffith called the mystery agent the transforming principle, but he did not identify it. Avery has spent years trying to isolate the transforming principle and determine what it is made of. Working with 2 colleagues, Colin MacLeod and Macklin McCarty, he systematically purifies the active component from the virulent bacteria. They treat it with proteases, enzymes that degrade proteins. The transforming activity remains. They treat it with lipases, enzymes that break down fats, still active. Finally, they treat it with an enzyme that specifically degrades DNA, deoxyribonuclease, or DNase. The transforming activity disappears. The conclusion is unavoidable. The transforming principle is DNA. Hereditary information can be transferred from one bacterium to another by purified DNA with no protein involved. Avery, MacLeod and McCarty published their findings in February 1944 in the Journal of Experimental Medicine. The paper is careful, thorough and understated. It does not claim to have discovered the chemical nature of the gene. It simply reports that DNA is capable of inducing a heritable change in bacterial cells. The response from the scientific community is cautious, skeptical, sometimes dismissive. The experiment is with bacteria, not with higher organisms. Perhaps bacteria are different. Perhaps the DNA is not acting as a gene itself, but merely activating a protein gene. Perhaps there are trace amounts of protein in the preparation that Avery's team missed, and those proteins are the real transforming agent. The decades-old conviction that proteins carry genetic information does not vanish overnight just because one experiment suggests otherwise. Avery himself is modest about the findings. He's in his 60s, a careful scientist who dislikes overstatement. He does not crusade for his result. He publishes it and moves on, but others are paying attention. At the Cold Spring Harbor Laboratory on Long Island, a geneticist named Alfred Hershey has been studying bacteriophages, viruses that infect bacteria. Phages are exquisitely simple. They are little more than a protein coat wrapped around a strand of DNA. When a phage infects a bacterium, it injects something into the cell, and that something hijacks the bacterium's machinery to produce hundreds of new phages. Hershey wants to know what gets injected, the protein coat or the DNA inside. In 1952, Hershey and his research assistant Martha Chase design an experiment using radioactive isotopes to label the 2 components separately. They grow phages in the presence of radioactive phosphorus-32, which gets incorporated into DNA but not into protein, because proteins do not contain phosphorus. They grow a second batch of phages in the presence of radioactive sulfur-35, which gets incorporated into protein but not into DNA, because DNA does not contain sulfur. Now they have 2 populations of phages, one with radioactive DNA, one with radioactive protein coats. They let each population of phages infect bacteria. Then, and this is the crucial step, they agitate the mixture in a Waring blender. The blender shears the empty protein coats off the surface of the bacteria without breaking the bacterial cells themselves. When they centrifuge the mixture, the bacteria pellet to the bottom, and the detached protein coats remain suspended in the liquid. Then they measure where the radioactivity is. The result is unambiguous. The phosphorus-32, which labeled the DNA, is inside the bacterial cells. The sulfur-35, which labelled the protein, is in the liquid, still attached to the empty coats. The DNA entered the cell. The protein coat did not, and the DNA alone was sufficient to direct the production of new phages. Hershey and Chase published their findings in 1952. The paper is direct, the figures are clear, and this time the scientific community listens. DNA is not structural scaffolding. It is not an inert framework. It is the message. It is the gene. The instructions for building a virus, and by extension the instructions for building any organism, are written in a 4-letter chemical alphabet, carried on a double-stranded molecule made of sugar, phosphate, and nitrogen bases. When a cell divides, that molecule is copied, and the copy is passed to the daughter cell. When a mistake is made during copying, that mistake is a mutation, a change in the instructions, and if the cell survives, the changed instructions are inherited by every cell that descends from it. The stakes are no longer abstract. An error in DNA replication is not merely a chemical accident, a misplaced atom in a complex molecule. It is a change in the blueprint for life. If the error occurs in a gene that controls cell division, the cell may begin to divide uncontrollably. If the error occurs in a gene that produces an essential protein, the protein may no longer function, and the organism may die. The fidelity of DNA replication is not a technical curiosity. It is the difference between an organism that functions and one that does not. It is the difference between life continuing and life ending. And now the question becomes sharper. DNA has been identified as the carrier of genetic information. It is known to be long, immensely long, when you consider that even a simple bacterium has millions of base pairs and a human cell has billions. It is known to be copied every time a cell divides, and the copy has to be accurate. Not perfect. Evolution depends on occasional errors, but accurate enough that the organism remains functional. How is that possible? What kind of molecular machinery can read a 6 billion letter message and reproduce it with an error rate of 1 in a billion? The answer will not come from genetics alone. It will require biochemistry, structural biology, and eventually the direct observation of the copying process itself. It will require discovering not just that DNA is the message, but how the message is written, how it is stored, and how it is maintained in the face of constant chemical assault. It will require understanding the double helix. The path to that understanding begins not with a single discovery, but with a race. By the early 1950s, the question of DNA structure has become one of the most urgent problems in all of science. Several research groups are working on it simultaneously, using different techniques, guided by different assumptions, driven by different motivations, and they are all aware of one another. In Cambridge, at the Cavendish Laboratory, 2 young researchers, James Watson, an American biologist, and Francis Crick, a British physicist turning toward biology, are building models, not conducting experiments, building models, physical structures made of metal plates and wire assembled on a workbench, adjusted and readjusted until the geometry matches the available data. Watson is 24, Crick is 35 and still working on his PhD. Neither is an expert in X-ray crystallography, the primary technique being used to probe molecular structure. But both are convinced that the structure of DNA will reveal the mechanism of heredity, and both are willing to speculate beyond what the data strictly allows. In London, at King's College, Rosalis Franklin is doing the opposite. She is generating the data. Franklin is a physical chemist and an X-ray crystallographer, trained in France, meticulous in method, cautious in interpretation. She does not build speculative models. She captures X-ray diffraction images of DNA fibers, measures the angles and intensities of the diffracted beams, and from those measurements calculates the positions of atoms within the molecule. It is slow work, it is rigorous work, and it is producing the clearest images of DNA structure anyone has yet obtained. Also at King's College is Maurice Wilkins, a physicist who has been working on DNA for several years, and who initially recruited Franklin to the project. The working relationship between Wilkins and Franklin is strained. There are misunderstandings about the division of responsibilities, differences in style, and unspoken tensions that will never be fully resolved. Wilkins shows some of Franklin's data to Watson. Franklin is not informed. And in California, Linus Pauling, one of the most accomplished chemists of the 20th century, is also building models. Pauling has already determined the structure of the alpha helix in proteins. He is confident, experienced, and moving quickly. In February 1953, He proposes a model for DNA, a three-stranded helix with the phosphate groups on the inside and the bases on the outside. The model is wrong. The chemistry does not work. The phosphates would repel each other. But Pauling does not yet know that, and for a few weeks Watson and Crick are terrified that he will correct the error before they can finish their own model. The breakthrough comes from an image, photograph 51, captured by Franklin and her PhD student Raymond Gosling in May 1952, using a technique called X-ray fiber diffraction. A beam of X-rays is directed at a DNA fiber, and the rays scatter off the atoms in the molecule. The scattered rays hit a photographic plate, creating a pattern of spots and smears. The pattern encodes the structure, if you know how to read it. Photograph 51 is the clearest image yet obtained of the B form of DNA, the form the molecule takes when it is hydrated, when it contains water molecules nestled along the helix. The exposure required 62 hours. The image shows a characteristic X shape, formed by the way the diffracted rays interfere with one another as they travel from a helical structure to the photographic plate. The X indicates a helix. The spacing of the spots indicates the pitch of the helix, the distance it takes for the structure to complete one full turn. Other features in the pattern constrain the diameter, the number of strands, and the arrangement of repeating units. Franklin interprets the image carefully. She calculates the helix parameters. She knows the structure is helical. She knows the phosphate groups are on the outside. She's working toward a full solution, testing different configurations, checking each against the data. She does not rush. Watson sees the photograph in January 1953 during a visit to King's College. Wilkins shows it to him. Franklin does not know. Watson does not ask for permission. He looks at the image, registers the X pattern, and returns to Cambridge. He tells Crick what he saw. Now Watson and Crick move faster. They already know from the chemical work of others that DNA contains equal amounts of adenine and thymine and equal amounts of guanine and cytosine. They know the molecule is long and likely helical. They know the bases are flat, ringed structures. And now they know, from Franklin's image, the precise geometry of the helix. 2 strands, not 3, wound around a common axis, with a full turn every 10 base pairs and a diameter that fits the data. They know the phosphates are on the outside, forming a sugar-phosphate backbone, because phosphates are negatively charged and would repel each other if packed together on the inside. That means the bases must be on the inside. But how do the bases pair? If the 2 strands are to be held together, something must link them. Watson tries pairing like with like, adenine with adenine, guanine with guanine. The geometry does not work. The distances are wrong. Then he tries pairing adenine with thymine and guanine with cytosine. Now the geometry fits. Each pair has the same width. Each pair can be held together by hydrogen bonds, weak chemical attractions between a hydrogen atom on one base and a nitrogen or oxygen atom on the other. Adenine and thymine form 2 hydrogen bonds. Guanine and cytosine form 3. The pairs are stable, they are specific, and they are complementary. Complementary. That is the word that changes everything. If adenine on one strand always pairs with thymine on the other, and guanine always pairs with cytosine, then the sequence of bases on one strand completely determines the sequence on the opposite strand. The 2 strands are not identical, they are mirrors, and that means if you separate the strands, each one carries the information needed to reconstruct the other. Watson and Crick build a model. Metal plates for the bases, rods for the sugar-phosphate backbone, everything held together with clamps. They adjust the angles. They check bond lengths and angles against known chemistry. They make sure the structure obeys the rules Franklin has derived from the data. The model stands on a laboratory bench, a double helix about 1 meter tall, elegant and simple. The 2 strands wind around each other in opposite directions. The bases pair across the center. The backbone spirals on the outside. On the morning of February 28, 1953, Crick walks into the Eagle, a pub near the Cavendish laboratory, and announces, according to Watson's later recollection, that they have found the secret of life. It is not modesty, but it is not entirely wrong either. They write up their findings. A short paper, fewer than 1,000 words, published in the journal Nature on April 25, 1953. The paper describes the structure, the 2 helical chains, the base pairing, the geometry. It is cautious in tone but clear in implication. The final sentence is famous, deliberately understated and pointed. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. A possible copying mechanism. If the two strands separate, each can serve as a template for building a new partner strand. Adenine on the old strand directs the placement of thymine on the new strand. Guanine directs cytosine, the sequence is preserved, the message is copied. On the same day, Nature publishes two more papers, one by Franklin and Gosling, presenting the X-ray data on the B form of DNA and confirming that the structure is helical. One by Wilkins and two colleagues, presenting data on the A form and noting consistency with the Watson-Crick model. The three papers together establish the double helix. The structure is accepted quickly, it fits the data. It explains the 1-to-1 ratios of bases, it accounts for the length and stability of the molecule, and it offers an immediate intuitive explanation for how genetic information could be copied. But, and this is the part that often gets lost, knowing the structure is not the same as knowing the mechanism. The double helix tells you what the template looks like. It does not tell you how the cell reads the template, or how it assembles the new strands, or how it ensures that the correct base is added each time. It does not tell you how the two strands are separated, or what force unwinds the helix, or how the new strands are joined together once the bases are in place. Watson and Crick's famous sentence says the structure suggests a copying mechanism. It does not describe the mechanism. That will take another decade, and there is something else the structure reveals, though it is not often put this way. The double helix is breathtakingly vulnerable. Each base pair is held together by hydrogen bonds which are weak. Heat can break them, chemicals can break them, even water under the right conditions can break them. The genetic message is written in a code that is chemically fragile, and the molecule carrying that code is exposed constantly to a cellular environment filled with reactive molecules, free radicals, and spontaneous chemical decay. The double helix is elegant, yes, but it is also under siege. Consider the numbers. Each human cell contains roughly 2 meters of DNA, not 2 meters total, distributed across the genome, 2 meters in a single cell. The human body contains approximately 37 trillion nucleated cells. If you took the DNA from all those cells and laid it end to end, the thread would stretch roughly 74 trillion meters. That is enough to reach from Earth to the Sun and back not once, not 10 times, but hundreds of times. It is a distance that defeats comprehension, and every single base pair in that 74 trillion meters is a potential site of error. Now bring the scale back down, back to a single cell, a single nucleus. The nucleus is a sphere, roughly 5 to 10 micrometers in diameter. A micrometer is 1/1,000,000th of a meter. You could fit 1,000 nuclei side by side across the width of a pinhead, and inside that nucleus, folded and coiled and twisted in ways that still are not fully understood, is 2 meters of DNA. 2 meters in 10 micrometers. The packing ratio is staggering. If you tried to stuff 2 meters of thread into a sphere 10 micrometers wide, you would fail. The thread would tangle, not break. The cell does not fail. The cell solves the problem with a hierarchical compression system that is one of the most remarkable pieces of molecular engineering in biology. Start with the DNA itself. The double helix, unadorned, has a diameter of about 2 nanometers. A nanometer is 1/1000,000th of a meter. The distance between adjacent base pairs, measured along the axis of the helix, is 0.34 nanometers. That is the first level of structure, the helix itself, the Watson-Crick double strand. Now the DNA wraps around proteins, histones, small, positively charged proteins, rich in the amino acids lysine and arginine, which carry positive charges that attract the negatively charged phosphate groups on the DNA backbone. Histones assemble into clusters, octamers, groups of 8, 2 copies each of 4 different histone proteins, H2A, H2B, H3, and H4. The octamer forms a spool, and the DNA wraps around it. 147 base pairs, wound 1 3/4 times around the histone core. This is the nucleosome, the fundamental repeating unit of chromatin. It compresses the DNA by a factor of roughly 6. The nucleosomes are spaced along the DNA like beads on a string, connected by short stretches of linker DNA. Another histone, H1, binds to the linker and pulls the nucleosomes closer together, stabilizing the structure. Now the beads on a string condense further, forming a fiber roughly 30 nanometers wide. The fiber coils, it loops, it folds back on itself, creating higher-order structures whose exact geometry is still debated, but the result is clear. The DNA is compressed by another factor of roughly 40. This is chromatin. the complex of DNA and histone proteins that fills the nucleus when the cell is not dividing. Chromatin exists in two primary states. Euchromatin, loosely packed, where the DNA is accessible and genes can be transcribed into RNA. Heterochromatin, densely packed, where the DNA is hidden and genes are silent. The cell toggles between these states by chemically modifying the histones, adding acetyl groups, which neutralize positive charges and loosen the grip on the DNA, or adding methyl groups, which can tighten it. Compression is not merely storage, it is regulation. The way the DNA is folded determines which genes are available to be read, but even this is not the final level of compression. When the cell prepares to divide, the chromatin condenses further. The loops tighten. Proteins called condensins bind to the DNA and pull distant regions together, forming thick, rod-shaped structures visible under a light microscope. These are chromosomes, 46 of them in a human cell, 23 pairs. Each chromosome is a single DNA molecule, coiled and compressed roughly 10,000-fold from its extended length. The concentration of DNA in a mitotic chromosome approaches millimolar levels, a density of nucleosomes so high that the structure is nearly crystalline. Walter Fleming saw these structures in the late 1870s, staining cells with dyes and watching the nucleus transform during cell division. He called the process mitosis. He described the thread-like metamorphosis of the nucleus, the chromosomes condensing, aligning at the center of the cell, separating into two groups, each group moving toward opposite poles. He did not know what the threads were made of. He did not know they carried genetic information, but he knew the threads were duplicated before division and that each daughter cell received an identical set. He was watching, without understanding, the preservation of the genome across generations. Now we know what he was watching. The genome compressed into 46 discrete packages, each package a single molecule of DNA wrapped around millions of nucleosome cores, the whole structure stabilized by condensins and topoisomerases, enzymes that pass one strand of DNA through another to relieve the torsional strain introduced by all that coiling. The compression is reversible. After division, the chromosomes decondense, the loops relax, the chromatin returns to its interphase state. But even in interphase, even when the chromatin is loosely packed, the DNA is not randomly arranged. Each chromosome occupies a distinct region of the nucleus, a territory. Chromosome 1 does not intermingle with chromosome 2. The genome is spatially organized, and that organization is maintained across cell divisions. Think about what this means for repair. Somewhere in that 2-meter coil, in a genome of 6 billion base pairs, a single base has been damaged. One cytosine has lost an amino group, converting it to uracil, which pairs with adenine instead of guanine. If the damage is not repaired before the next replication, the error will be copied and the mutation will be permanent. The cell has to find that single damaged base among 6 billion. It has to find it while the DNA is wrapped around histones, while the chromatin is folded into loops, while the chromosomes are confined to territories. It has to unwind the nucleosome, expose the damaged region, cut out the incorrect base, fill in the correct one, and reseal the backbone. And it has to do this thousands of times a day because DNA damage is not rare, it is constant. The double helix promised a copying mechanism. Separate the strands, use each as a template, rebuild the complement, simple in principle, but the principle assumes the strands can be separated. It assumes the template is accessible. It assumes the machinery can move along the DNA without getting tangled in the histones, without distorting the loops, without breaking the strands, and it assumes the machinery operates with extraordinary fidelity because the cost of error is mutation, and the cost of uncorrected mutation is disease, dysfunction, death. The elegance of the structure has created a new mystery. How does the cell execute the copy? How does it open the double helix, hold the strands apart, read the template, add the correct bases, and seal the new strands together, all at a speed that allows a human cell to copy 3 billion base pairs in roughly 8 hours? How does it compress 2 meters of DNA into a nucleus 10 micrometers wide and still find a single damaged base among billions? The structure is known. The problem is defined. The answer will require looking closer at the enzymes that unwind the helix, closer at the proteins that hold the strands apart, closer at the polymerase that builds the new strand, one base at a time, checking each addition before moving on. The double helix is the map. Now we have to watch the machinery move. The fork opens in the middle of the genome like a Y laid on its side, the two template strands peeling apart, exposing the bases that have been paired and protected since the last division. This is where the copying begins, not everywhere at once, but at tens of thousands of discrete starting points scattered across the chromosomes. Each origin fires in a defined sequence, and from each origin a replication fork moves in both directions, unwinding the helix, separating the strands, building two new daughters where there was one parent. The fork is not a static structure. It is a moving machine, a coordinated assembly of proteins advancing along the DNA at roughly 30 to 50 bases per second in a human cell. Slower than in bacteria, where the fork can move 10 times faster, but fast enough that the entire human genome, 3 billion base pairs, can be copied in about 8 hours, the length of S phase in a typical dividing cell. At the front of the fork sits helicus, a motor protein that consumes ATP and uses the energy to break the hydrogen bonds between paired bases, forcing the double helix to unwind. The helicus does not simply pull the strands apart, it walks along one strand, gripping it, ratcheting forward one base at a time, and as it moves, the helix rotates ahead of it, the coils tightening into positive supercoils that must themselves be relieved by toposomerases, enzymes that cut one or both strands temporarily, allow the coil to relax and reseal the break. Without toposomerases, the fork would stall, the tension ahead of the helicase building until the DNA itself twisted into knots. With them, the fork moves steadily, exposing perhaps a dozen bases at a time, a short single-stranded bubble held open by proteins that coat the exposed template and prevent it from snapping back into a helix before the polymerase arrives. DNA polymerase is the enzyme that adds new nucleotides, but it cannot start from nothing. It requires a short stretch of paired bases, a primer, to provide the free 3' hydroxyl group it needs to form the next phosphodiester bond. That primer is synthesized by primase, an enzyme that lays down a short hybrid chain of RNA and DNA, 10 to 35 bases long, giving the polymerase a foothold. Once the primer is in place, the polymerase takes over, reading the template strand and adding the complementary base, adenine opposite thymine, guanine opposite cytosine, extending the new strand in the only direction chemistry allows, 5' to 3'. Here is where the asymmetry begins. The helicase is moving in one direction, unwinding the helix as it goes, but the two template strands run in opposite directions. One strand, the leading strand, is oriented so that the polymerase can follow the helicase continuously, adding bases in the same direction the fork is moving. The other strand, the lagging strand, runs the wrong way. The polymerase cannot move backward. It can only add nucleotides 5' to 3', which means it must work away from the fork, synthesizing in short bursts, each burst beginning with a new primer laid down by primase as the helicase opens more template. These short segments are called Okazaki fragments, named for the Japanese biochemists Reiji Okazaki and Tsuneko Okazaki, who presented the first clear evidence for discontinuous DNA synthesis at Cold Spring Harbor in 1968. In human cells, each Okazaki fragment is roughly 100 to 200 bases long. In bacteria, they stretch longer, 1,000 to 2,000 bases. The difference is not arbitrary. It reflects the spacing of primers, the speed of the polymerase, the distance the fork travels before another primer is needed. On the lagging strand, the polymerase synthesizes one fragment, releases, waits for the next primer, begins again. The leading strand, by contrast, is copied in a single unbroken run from the origin to the end of the replicon, thousands or millions of bases without pause. Two different polymerases handle the two strands in eukaryotic cells. Polymerase epsilon typically synthesizes the leading strand. Polymerase delta synthesizes the lagging strand, and recent evidence suggests it can also proofread errors made by polymerase epsilon, a backup system called extrinsic proofreading. Both enzymes are large, multi-subunit machines, each carrying not only the catalytic core that adds nucleotides, but also an exonucleus domain that removes them. This is the first line of fidelity. The polymerase itself can reverse, inspect what it has just added, and excise a mistake before moving on. The error rate without proofreading would be roughly one mistake in every 100,000 bases added. That sounds precise until you multiply it by 3 billion. A single round of replication would introduce 30,000 errors into the genome. Most would be harmless, falling in non-coding regions or altering a protein in a way that does not affect its function. But some would land in essential genes, disrupting regulation, truncating enzymes, breaking the machinery the cell depends on. 30,000 errors per division is not sustainable. Life at this scale, with genomes this large, requires something better. The polymerase achieves that by checking each added nucleotide before committing to the next. A correct base pair, adenine with thymine, guanine with cytosine, fits the helix geometry exactly. The hydrogen bonds align, the spacing is uniform, the sugar-phosphate backbone remains smooth. A mismatch distorts that geometry. If the polymerase tries to add a guanine opposite a thymine, the pair is too narrow. If it adds an adenine opposite a guanine, the pair is too wide. The distortion is subtle, a fraction of an angstrom, but it stalls the polymerase. The active site, which is shaped to accommodate a correctly paired base, cannot easily extend a mismatched one. The polymerase pauses. That pause gives the exonuclease time to act. The exonucleus domain is located about 40 angstroms away from the site where new bases are added. When the polymerase stalls, the DNA can shift from the polymerase active site into the exonucleus site, where the 3' end of the newly synthesized strand is cleaved, removing the last nucleotide. The polymerase then shifts the DNA back, tries again, and if the correct base is added this time, the geometry is right, and synthesis continues. This process, adding a base, checking the fit, reversing if the fit is wrong, reduces the error rate by roughly 100-fold. The intrinsic error rate of 1 in 100,000 becomes 1 in 10 million after proofreading. That is still not 1 in a billion. There is one more inspection to come, and it happens after the replication fork has passed, after the new strand has been synthesized, after the Okazaki fragments have been joined and the primers replaced with DNA and the nick sealed by ligase. The third inspection is called mismatch repair. A polymerase operating at 50 bases per second, even with proofreading, will let one error slip through for every 10 million bases it synthesizes. In a genome of 3 billion base pairs, that means roughly 300 mismatches per cell division escape the polymerase. 300 places where a guanine sits across from a thymine, or an adenine across from a cytosine, a pairing that should not exist, a subtle wrongness embedded in a newly built strand. If those mismatches are not corrected, they will be locked in at the next replication, one strand reading the template as it was meant to be, the other reading the error as though it were truth. Mismatch repair finds those errors, it scans the newly replicated genome, identifies the mismatches, determines which strand is the template and which is the new copy, excises a stretch of the new strand around the error, and rebuilds it correctly. The system relies on a set of proteins whose names, MSH2, MSH6, MLH1, PMS2, are now familiar to clinicians because mutations in the genes that encode them cause cancer. The mechanism was worked out first in bacteria, where the system could distinguish the old strand from the new by detecting methylation marks that took time to appear on the daughter strand. In eukaryotes, the marks are different. The newly synthesized strand is identified by transient nicks left behind by the replication machinery, specifically at sites where the sliding clamp protein pCNA is loaded onto the DNA to tether the polymerase. The mismatch repair proteins scan the genome, and when they encounter a mismatch, they recruit an endonuclease, usually the MLH1-PMS2 complex, that cuts the new strand near the mismatch. An exonuclease then chews back the strand, removing not just the mismatched base but a stretch of surrounding sequence, sometimes 1,000 bases or more. DNA polymerase delta fills in the gap, using the intact template strand as a guide, and ligase seals the nick. The system is fast and nearly infallible. It reduces the final error rate to somewhere between 1 mistake in a billion bases and 1 in 10 billion, depending on the measurement and the cell type. That is the figure that allows complex multicellular organisms to exist, to develop from a single fertilized egg into trillions of differentiated cells, to maintain those cells across decades, to pass the genome to the next generation with enough fidelity that the offspring resemble the parents, but enough variation that evolution can continue. When mismatch repair fails, the error rate climbs back toward 1 in 10 million. That is a 100-fold increase, and it is enough to flood the genome with mutations. In bacterial cells, loss of mismatch repair produces a mutator phenotype. Populations that evolve rapidly, adapting to new environments but also accumulating deleterious changes that eventually limit growth. In human cells, the effect is more severe. A single cell that loses mismatch repair does not immediately die. It replicates, and each daughter cell carries the same defect, and the daughters replicate, and within a few divisions the lineage has accumulated hundreds of new mutations, scattered across the genome, most of them silent but a few landing in genes that control cell division, apoptosis, DNA repair itself. This is Lynch syndrome, named for the American physician and geneticist Henry Lynch, who spent decades from the 1960s onward documenting families in which colorectal and endometrial cancers appeared generation after generation, far more often and far younger than chance would predict. The pattern was clear, but the mechanism was hidden until the 1990s, when researchers identified mutations in the mismatch repair genes MSH2, MLH1, PMS2, MSH6 as the cause. Individuals carrying a germline mutation in any of these genes inherit one defective copy in every cell. The other copy, inherited from the unaffected parent, is functional, and mismatch repair proceeds normally as long as that copy remains intact. But if a second mutation occurs in a single cell, knocking out the remaining functional copy, that cell loses mismatch repair entirely. The path from there to cancer is not immediate, but it is nearly inevitable. The cell replicates, the error rate climbs, mutations accumulate in genes that regulate the cell cycle, tumor suppressors, oncogenes, the genes that tell a cell when to divide and when to stop. The cell begins to ignore the signals that constrain it. It divides when it should not, survives when it should die, and eventually forms a tumor. The lifetime risk of colorectal cancer in Lynch syndrome ranges from 33% to 80%, depending on which gene is mutated. The risk of endometrial cancer ranges from 21 to 60%. Other cancers, ovarian, gastric, pancreatic, are also elevated. The first documented family was recorded in 1895 by a pathologist named Aldred Scott Warthin at the University of Michigan. His seamstress, a woman named Pauline Gross, told him she expected to die young of cancer because everyone in her family did. Warthin was intrigued, and over the next 2 decades he traced the family's medical history, documenting case after case of uterine and abdominal cancers across multiple generations. He published the pedigree in 1913 in the Archives of Internal Medicine, calling it Family G. The paper was largely ignored. The idea that cancer could be inherited, that it could follow Mendelian patterns, was not widely accepted. It would take another 50 years, and the work of Henry Lynch and others, before the genetic basis of the syndrome was understood, and another 2 decades before the responsible genes were identified. Lynch syndrome accounts for 2 to 5% of all colorectal cancers and 2 to 3% of all endometrial cancers. It affects roughly 1 in 300 people in the general population, though the prevalence varies by ancestry and region. Most carriers do not know they carry the mutation until a tumor appears, often in their 30s or 40s, decades earlier than sporadic cancers typically arise. The tumors can be detected early through regular screening, and in many cases they can be removed before they metastasize, but the underlying defect remains. The cells in the affected tissue continue to accumulate mutations and new tumors can form. The syndrome is a window into the importance of the spell checker. Mismatch repair is not the only line of defense. There is the polymerase's proofreading. There are other repair pathways that correct different kinds of damage. But it is the last check on replication errors, the final inspection before a mismatch becomes a mutation. When that inspection fails, the genome becomes unstable, and instability is the engine of cancer. Let that settle for a moment. The replication fork moves at 50 bases per second, copying 3 billion base pairs in 8 hours. The polymerase checks each base as it is added, rejecting mismatches, pausing to correct errors. The mismatch repair system scans the newly synthesized strand, finds the errors that slipped through, cuts them out, rebuilds the sequence. 3 layers of inspection, each one reducing the error rate, each one necessary. And still, even with all 3 layers functioning, roughly 1 mutation per cell division escapes into the genome. 1 error in 1 billion bases. That is enough to drive evolution over geological time, enough to introduce variation into populations, enough to allow adaptation, but it is also enough to cause disease if the error lands in the wrong place. The cell cannot eliminate errors entirely. The chemistry of base pairing is not perfect. Thymine and cytosine are similar enough in shape that a polymerase, moving at speed, will occasionally place one where the other belongs. Purines and pyrimidines can adopt rare tautomeric forms, temporary shifts in their chemical structure that allow incorrect pairs to fit the helix geometry well enough to be accepted. And DNA itself is not stable. Bases are lost spontaneously, hydrolyzed off the sugar phosphate backbone, Cytosines lose their amino groups, converting to uracil. Adenines and guanines are lost entirely, leaving a sugar with no base attached, a gap the polymerase cannot read. These changes happen constantly, even in the absence of external damage, driven by the thermal energy of the cell, by stray reactive molecules, by the simple fact that chemical bonds are not eternal. The replication fork moving through this landscape is not just a copying machine, it is a rescue operation, a continuous attempt to preserve the message against the erosion of chemistry, and the speed matters. The longer the DNA remains single-stranded, the more vulnerable it is. Single-stranded DNA is more chemically reactive than double-stranded, more easily damaged, more prone to breaking. The replication fork is a site of tension, the helicase pulling the strands apart, the polymerase racing to cover them again, and if the two become decoupled, if the helicase moves ahead and the polymerase falls behind, the fork stalls. The exposed single-stranded region grows. Proteins are recruited to stabilize the structure, to prevent collapse, but a stalled fork is a fragile thing, a place where the genome is most at risk of breaking. When the fork stalls, the cell responds by activating a checkpoint, a signaling pathway that halts the cell cycle and gives the repair machinery time to resolve the problem. The checkpoint is mediated by a protein kinase called ATR, which is recruited to stalled forks and phosphorylates downstream targets, triggering a cascade that stops further DNA synthesis and activates repair pathways. If the fork cannot be restarted, if the damage is too severe, the checkpoint remains active and the cell either repairs the damage or, if repair fails, initiates apoptosis, programmed cell death. Better to lose one cell than to allow a broken genome to replicate. The fork is fragile, the machinery is fast but not invincible, and the error rate, even with 3 layers of correction, is not zero. The genome is copied with extraordinary fidelity, but fidelity is not perfection. 1 error per billion bases, 1 mutation per cell division. Over a human lifetime, over trillions of cell divisions, that adds up. Most mutations are harmless, landing in regions of the genome that do not code for proteins, or altering proteins in ways that do not affect their function. But some land in places that matter, and when the repair systems fail, when the proofreading is defective or the mismatch repair is lost, the error rate climbs and the mutations accumulate and the cell begins to change. This is the cost of replication. The double helix promised a mechanism for copying, and the cell has built extraordinary machinery to execute that mechanism with precision. But the machinery operates in a dynamic, chemically reactive environment, under time constraints, under mechanical tension, and the perfection it achieves is not absolute. It is good enough. It is good enough to preserve the message across generations, good enough to allow an organism to develop and live and reproduce, good enough to limit the number of errors to a level that evolution can tolerate. But it is not good enough to prevent all damage, and the damage that remains is where the next problem begins. Because the problem is not only the copying. Every cell in your body is under continuous assault, and the attack began long before the replication fork ever opened. Water is the first threat. The same molecule that makes life possible is, in chemical terms, corrosive. DNA sits in an aqueous environment, bathed in the cytoplasm, and water molecules collide with the bases constantly. Sometimes a collision is enough. A water molecule reacts with a chemical bond in the DNA and the bond breaks. This process is called hydrolysis, and it happens spontaneously, driven by thermal energy, by the simple fact that the cell is warm. One of the most common targets is the glycosidic bond, the link between the nitrogenous base and the sugar backbone. When that bond breaks, the base is lost entirely. What remains is an AP site, a purinic or a pyrimidinic, a sugar with nothing attached, a blank space in the message. Another common target is cytosine. Cytosine contains an amino group, a chemical feature that makes it unstable in water. The amino group can be hydrolyzed off, converting cytosine into uracil. Uracil belongs in RNA, not DNA, and its presence in DNA is a form of damage, a change in the message that, if left unrepaired, will be copied as a mutation in the next round of replication. The rate is not slow. In a single human cell, thousands of bases are lost to hydrolysis every day. Thousands of cytosines are converted to uracil every day, and this is in the absence of any external damage, in the dark, at body temperature, inside a cell that is otherwise functioning normally. Then there is oxygen. Oxygen is essential for energy production, but it is also a chemical threat. Cellular respiration generates reactive oxygen species, superoxide, hydrogen peroxide, hydroxyl radicals, molecules that are chemically aggressive, that will oxidize nearly anything they encounter. DNA is a target. The bases, particularly guanine, are vulnerable to oxidation. When a guanine is oxidized, it becomes 8-oxoguanine, a modified base that can mispair with adenine instead of cytosine, leading to a mutation if the damage is not repaired before replication. The sugar phosphate backbone is also vulnerable. Oxidative damage can break the backbone entirely, creating a strand break, a discontinuity in the helix that is far more dangerous than a damaged base. The human body produces antioxidants to neutralize reactive oxygen species, but the defense is not perfect. The damage accumulates. Estimates vary, but a reasonable figure is that each cell sustains tens of thousands of oxidative lesions every day. Tens of thousands of chemical modifications to the genome, happening continuously as a side effect of breathing. Light is another assailant. Ultraviolet light, the component of sunlight that causes sunburn, carries enough energy to break chemical bonds. When UV light is absorbed by DNA, it can cause two adjacent thymine bases to form a covalent bond with each other, creating a structure called a thymine dimer. The dimer distorts the helix, kinking the DNA, blocking the polymerase during replication. If the damage is not repaired, the replication fork stalls, and the mutation that results is usually a deletion or a frameshift, both of which can destroy the function of a gene. Longer wavelengths, UVA light, generate reactive oxygen species, adding to the oxidative burden. The skin is the most exposed tissue, but UV light penetrates windows, penetrates clouds, penetrates the atmosphere even on overcast days. The damage is cumulative. Chemical exposure adds to the load. Tobacco smoke contains at least 60 known carcinogens, aldehydes, nitrosamines, polycyclic aromatic hydrocarbons, all of which can form adducts with DNA, bulky chemical groups attached to the bases that distort the helix and block replication. Alcohol is metabolized into acetaldehyde, which is genotoxic. Charred food contains heterocyclic amines, industrial solvents, pesticides, preservatives, plastics. Each brings its own roster of chemicals that can, in sufficient concentration, react with DNA. Even foods contain naturally occurring mutagens. The environment is not neutral, the genome is chemically exposed, and then there is the background hum of radiation. Cosmic rays, originating from supernovae and other high-energy events in deep space, pass through the Earth's atmosphere and through your body continuously. Most pass without incident, but occasionally a particle will collide with an atom in a DNA molecule, ionizing it, breaking the backbone, creating a double-strand break. Radon gas, a decay product of uranium naturally present in soil and rock, accumulates in basements and ground floors, releasing alpha particles that, though they travel only short distances, carry enough energy to shatter DNA when they strike. Medical imaging, CT scans, X-rays, add to the cumulative dose. The level is low, but it is never zero. The genome, then, is not a pristine archive. It is a manuscript left open on a desk in a room with an open window, in sunlight, in the rain, in a place where the air itself is mildly acidic, and the paper is slowly turning yellow. And people are still reading it, still copying it, still trying to use it to build proteins, all while the ink fades and the edges curl and whole sentences dissolve into the margins. For decades, the working assumption was that DNA must be chemically stable. It had to be. How else could the genetic message be preserved across generations, across millions of years of evolutionary time? The double helix was described in 1953, its structure revealing a molecule that appeared elegant, symmetric, chemically balanced. It seemed reasonable to assume that a molecule carrying such an important message would be inherently stable, protected by its structure from the ordinary wear of chemistry. Thomas Lindahl did not believe it. Born in Stockholm in 1938, trained as a biochemist, Lindahl began working on DNA in the late 1960s, and from the beginning he was skeptical. He had been trained in the chemistry of nucleic acids, and he knew that the bases were not chemically inert. Cytosine in particular should be unstable in water. The amino group should come off. The textbook said DNA was stable, but the chemistry said otherwise, and Lindahl trusted the chemistry. He began to measure it. Working in Sweden, and later in the United States, he took purified DNA and incubated it at body temperature, in neutral pH, in the absence of enzymes or cells, and he watched it decay. Bases were lost. Cytosines were converted to uracil. The rate was slow by human standards but fast by cellular standards. He calculated that, left unrepaired, the spontaneous decay of DNA would destroy the genome within a few generations. The molecule was not stable, it was fragile, and yet life persisted. The only explanation was repair. The genome was not preserved by being indestructible. It was preserved by being continuously repaired, damage detected and excised and replaced, faster than it accumulated. Lyndall began to search for the machinery. He moved to Clare Hall Laboratories, a research facility in Hertfordshire, and spent decades characterizing the enzymes that recognize damaged bases, that cut them out, that fill in the gap, that restore the original sequence. He was not working alone. By the 1970s, laboratories around the world were beginning to realize that DNA repair was not an occasional emergency response, but a continuous, essential process, running in every cell, in every organism, at all times. One of the pathways Lyndall characterized was base excision repair. It is the system the cell uses to fix damage to individual bases, oxidized guanines, deaminated cytosines, alkylated adenines. The pathway begins with a glycosylase, an enzyme that patrols the DNA, recognizing a specific type of damaged base. The glycosylase flips the damaged base out of the helix, inspects it, and if it is damaged, cleaves the glycosidic bond, removing the base entirely and leaving an AP site. Then, an AP endonuclease recognizes the AP site and cuts the sugar phosphate backbone, creating a single-strand break. A polymerase fills in the missing base, using the opposite strand as a template, and a ligase seals the nick. The pathway is fast, efficient, and highly specific. The cell has multiple glycosylases, each recognizing a different type of damage. One for uracil, one for atoxoguanine, one for alkylated bases. The system is built on redundancy and specialization. Lyndall was awarded the Nobel Prize in Chemistry in 2015, shared with two other researchers, for his work on DNA repair. The citation noted that his discoveries had overturned the prevailing view of DNA as a stable molecule and revealed that life depends not on the absence of damage, but on the continuous correction of it. But base excision repair is only one pathway. Some forms of damage are too large, too bulky, too distorting to be handled by removing a single base. For those, the cell uses nucleotide excision repair, a system that cuts out not just the damaged base but an entire stretch of DNA, a segment 20 to 30 nucleotides long, and replaces it wholesale. The classic trigger for nucleotide excision repair is ultraviolet light. When UV light creates a thymine dimer, the dimer kinks the helix, and the kink is what the repair machinery recognizes. A complex of proteins scans the DNA, detecting distortions in the helix geometry. When a distortion is found, the complex recruits an endonuclease, an enzyme that makes two cuts, one on each side of the damage, excising the damaged strand as a short single-stranded fragment. A polymerase fills in the gap, using the undamaged strand as a template, and a ligase seals the ends. The pathway is slower than base excision repair, more complex, involving more proteins, but it can handle a wider range of damage. It repairs not just thymine dimers but any bulky adduct, any chemical modification that distorts the helix enough to be detected. The importance of nucleotide excision repair became clear in 1968, when a biochemist named James Cleaver, working at the University of California, San Francisco, published a short paper in the journal Nature. Cleaver had been studying a rare genetic disorder called xeroderma pigmentosum. People with xeroderma pigmentosum cannot tolerate sunlight. Even brief exposure causes severe sunburn, blistering, freckling. By adolescence, most develop skin cancer, often multiple cancers, in sun-exposed areas. The condition had been described clinically in the 19th century, but the molecular cause was unknown. Cleaver had read about DNA repair in bacteria, and he wondered whether the human disorder might involve a defect in the same process. He obtained skin cells from patients with xeroderma pigmentosum and exposed them to UV light and culture. Normal cells, after UV exposure, would synthesize new DNA as part of the repair process, replacing the damaged segment. Cleaver measured the DNA synthesis in the patient's cells. It was absent. The cells could not repair UV damage. The link was direct. A defect in DNA repair caused extreme sensitivity to sunlight and a massive increase in cancer risk. It was the first time a human genetic disease had been traced to a defect in a specific DNA repair pathway. The disorder is rare, affecting roughly 1 in a million people, but it is not uniform. There are multiple genes involved in nucleotide excision repair, and mutations in any of them can cause xeroderma pigmentosum. The genes are named XPA through XPG, each encoding a different component of the repair machinery. Some forms of the disorder are severe, with cancer appearing in early childhood. Others are milder, with a later onset. But all share the same fundamental problem. The machinery that removes bulky DNA lesions is broken, and the damage accumulates, and the mutations follow. Philip Hanawalt, working at Stanford, had been studying DNA repair in bacteria since the 1950s and was among the first to demonstrate that cells could repair UV-induced damage. His work, and the work of others in the 1960s and 1970s, established nucleotide excision repair as a conserved pathway, present in bacteria, in yeast, in humans, one of the most ancient and essential systems in biology. The cell has yet another class of repair systems, rarer but elegant: direct reversal. Instead of cutting out the damage and replacing it, these pathways chemically reverse the damage, restoring the base to its original state without removing it. One example is photolysis, an enzyme that recognizes thymine dimers and, in the presence of visible light, breaks the covalent bond between the two thymines, restoring the original bases. Humans do not have photolysis, we lost it somewhere in mammalian evolution, but many organisms do, and it remains one of the most efficient repair pathways known. Another example is a protein that removes methyl groups added to guanine by certain carcinogens, directly reversing the alkylation. These pathways are narrow in scope, each handling only one type of damage, but where they apply, they are fast and error-free. The result is a system of extraordinary redundancy. Over 130 proteins are known to be involved in DNA repair, each with a specific role, each monitoring a specific kind of damage. The pathways overlap. A single lesion might be recognized by multiple systems. The cell prioritizes. Damage in actively transcribed genes is repaired faster than damage in silent regions of the genome. Damage on the template strand is repaired faster than damage on the coding strand. The system is not egalitarian, it is triage. The most critical parts of the genome are protected first, and the rest is handled as resources allow, and the need is constant. Estimates vary depending on the method of measurement and the tissue being studied, but a reasonable figure is that each cell sustains between 10,000 and 100,000 DNA lesions every day. 10,000 AP sites, 10,000 oxidized bases, 10,000 small distortions, breaks, chemical modifications, every day, in every cell. Across 37 trillion cells, that is a number almost too large to hold. The genome is not a finished document. It is a document being written and erased and rewritten continuously, and the fact that it remains readable at all is not due to its stability, but to the vigilance of the repair machinery, working without rest in every cell in every tissue from the moment of conception until death. Cleaver's discovery, and Lindahl's and Hanawalt's, overturned the myth of DNA as an eternal molecule. The double helix is not a vault. It is a battlefield, and the custodians of the genome are not guards standing at the entrance. They are medics, running through the wreckage, patching what can be patched, replacing what cannot, working faster than the damage arrives, and just barely winning. Because when the repair systems fail, when a gene is mutated, when the machinery is missing or overloaded, the damage is no longer temporary. It becomes permanent. It is copied during replication, transmitted to daughter cells, inherited by the next generation. And if the damage lands in the wrong place, in a gene that controls cell growth, or a gene that triggers apoptosis, or a gene that itself encodes a repair protein, the consequences compound. One error leads to more errors. One broken system makes the others less effective. The cell begins to drift, its genome accumulating mutations faster than they can be fixed, and the balance that allowed it to survive tips towards something else. The body's repair systems handle base damage, distorted letters, breaks in a single strand. But there is another kind of injury, rarer and more dangerous, when both strands break at once. A double-strand break is not a typo. It is a severed chromosome, split across its width, both rails of the ladder cut. The break may come from ionizing radiation, a gamma ray from medical imaging, a cosmic particle that arrived after crossing the galaxy. It may come from a replication fork colliding with a lesion. The repair systems missed, the fork stalling and collapsing, the stress fracturing the DNA. Or it may be deliberate, cut by enzymes during a controlled process the cell requires, and the danger accepted as necessary. Most mammalian cells sustain a handful of double-strand breaks every cell cycle, a small but unavoidable consequence of copying and maintaining the genome. The frequency climbs higher in cells that divide rapidly or perform specialized genomic rearrangements. If the break is not repaired, the chromosome becomes unstable. Its ends may fuse with other chromosomes, forming aberrant structures that cannot segregate properly during cell division. Or the fragment may be lost entirely, taking with it hundreds or thousands of genes. Either outcome is catastrophic. Double-strand breaks are one of the most lethal lesions a cell can sustain. They are also among the hardest to fix. The cell has two main strategies for repairing them, and the choice between them is a choice between speed and fidelity. The first pathway is called non-homologous end joining. The name is technical but descriptive. The cell joins the two ends without reference to a template, without checking what was originally there. The break is recognized by a protein complex that binds to the severed ends and holds them in proximity. Enzymes trim the frayed DNA strands, clean up the ends, fill in any gaps with new nucleotides, and ligate the pieces back together. The whole process takes roughly 30 minutes. It is the dominant repair pathway in human cells, active throughout the cell cycle, efficient and fast. It is also mutagenic. Because the repair machinery does not consult a template, it cannot restore the original sequence. Small insertions or deletions, indels, are common. A few nucleotides may be added or lost at the break site. If the break occurred within a gene, the reading frame may shift, the protein sequence scrambled. If it occurred in a regulatory region, the gene's expression may change. Non-homologous end joining keeps the chromosome intact, but it does so at the cost of precision. The scar remains. The second pathway is slower and more accurate. It is called homologous recombination. Homologous recombination uses the sister chromatid as a template. After replication, each chromosome exists in two identical copies, held together at the centromere until cell division. If a double-strand break occurs after replication, the cell can use the undamaged sister as a reference, copying its sequence to restore the broken strand. The fidelity is high. The original sequence is preserved, but the process is complex, requiring dozens of proteins working in sequence, and it takes hours, 7 or more in many cases, to complete. And it is only available during a narrow window of the cell cycle, after replication has occurred and before the sister chromatids separate. At the heart of homologous recombination are two proteins known as BRCA and BRCA. The names are abbreviations: breast cancer susceptibility gene 1, breast cancer susceptibility gene 2, and they encode what the names suggest. But their function is broader than the label implies. Both proteins are tumor suppressors, yes, but their primary role is in DNA repair. They are scaffolding proteins, recruiting and organizing the dozens of other factors required for homologous recombination. Without them, the pathway cannot proceed. The broken DNA is recognized, but the machinery cannot assemble. The repair stalls. The gene BRCA1 was mapped in 1990 by Mary Claire King, a geneticist who had spent years analyzing families with high rates of early-onset breast cancer, searching for the genetic signature linking them. The gene was identified on chromosome 17. BRCA2 followed in 1994, mapped to chromosome 13 by a collaborative effort involving multiple research groups. Both genes are large, encoding proteins with thousands of amino acids, and both are essential. Women who inherit a pathogenic variant in BRCA1 or BRCA2 face a lifetime breast cancer risk between 55 and 80%, compared to roughly 13% in the general population. Ovarian cancer risk is similarly elevated. The numbers vary depending on the specific mutation, the population studied, and the surveillance methods, but the pattern is consistent. The genes were named for the cancers they predispose to, but the cancers arise because the repair pathway is broken. A cell with one functional copy of BRCA1 or BRCA2 can usually manage. The proteins are abundant and one working gene produces enough. But if the second copy is lost, through mutation, through deletion, through any of the mechanisms that can silence a gene, the cell is left with no functional BRCA protein. Homologous recombination fails. Double-strand breaks accumulate, the genome destabilizes, mutations pile up, and among them, eventually, are mutations in genes that control cell growth. The cell begins to divide uncontrollably. It becomes a tumor. The pattern was observed long before the genes were known. In 1866, a French surgeon named Pierre-Paul Broca documented breast cancer across 3 generations of his wife's family. The disease appeared repeatedly, early, in multiple members. The inheritance was clear. What was not clear for more than a century was the mechanism. The identification of BRCA1 and BRCA2 answered that. The genes were the first hereditary cancer genes to be cloned from humans, and their discovery reshaped both cancer genetics and the clinical management of familial cancer risk. But the discovery also opened another door. If a cancer cell loses BRCA function, it becomes dependent on other repair pathways to survive. It cannot use homologous recombination, so it relies more heavily on non-homologous end joining and on pathways that handle single-strand breaks. One such pathway involves a protein called PARP, poly-ADP ribose polymerase, which detects and helps repair single-strand breaks. PARP is essential in normal cells, but in BRCA-deficient cells, it becomes even more critical. The cells are already fragile, their genomes unstable, and PARP is one of the few remaining lifelines. This creates a vulnerability. If PARP is inhibited, single-strand breaks go unrepaired. During replication, the fork encounters these breaks and collapses, creating double-strand breaks. In a normal cell, homologous recombination would step in and repair them, but in a BRCA-deficient cell, homologous recombination is already broken. The double-strand breaks cannot be repaired. The damage is lethal. The cell dies. This is called synthetic lethality. Neither defect alone is fatal. Cells can survive without PARP inhibition, and they can survive without BRCA, but together the two defects are incompatible with life. The concept had been understood theoretically for decades, studied in yeast and fruit flies, but translating it into therapy required identifying the right drug and the right target. PARP inhibitors were developed in the early 2000s. Olaparib, the first to reach clinical approval, was authorized for use in BRCA-mutated ovarian cancer in 2014. Others followed: rucaparib, niraparib, talazoparib. The drugs are now approved for breast, ovarian, prostate, and pancreatic cancers with BRCA mutations. They are not cures. Resistance develops. But in many patients, they achieve durable responses, shrinking tumors that had stopped responding to chemotherapy, buying time measured in years. The mechanism is elegant. The drug does not kill the cancer cell by attacking it directly. It kills the cell by removing a crutch the cell itself created when it lost BRCA. The therapy exploits the tumor's own repair deficiency, and it works because the repair machinery is not a monolith. It is a network of interdependent pathways, and breaking one pathway makes the others more important. The redundancy that protects the genome can also become a weakness, but not all double-strand breaks are accidents. In some cells, at certain stages of development, the genome is cut deliberately. The most striking example occurs in the immune system, in the development of B cells and T cells, the lymphocytes responsible for recognizing and responding to pathogens. Each B cell produces antibodies. Each T cell carries receptors that recognize foreign molecules. The diversity of these receptors is staggering, billions of unique combinations in a single human being. And that diversity is generated not by encoding billions of separate genes, but by cutting and recombining a small number of gene segments in different combinations. The process is called VDJ recombination, named for the three classes of gene segments involved: variable, diversity, and joining. In a developing B cell, the segments are scattered along the chromosome. Enzymes cut the DNA at specific sites flanking the segments, creating double-strand breaks. The cell then selects one segment from each class and joins them together, discarding the intervening DNA. The result is a unique combination that encodes a unique antibody. The same process occurs in T cells, generating receptors. The enzymes that perform the cutting are called RAG1 and RAG2, recombination activating genes 1 and 2. They were identified in 1989 and 1990, cloned from mouse and human lymphocytes, and shown to be necessary and sufficient for initiating VDJ recombination. The RAG proteins form a complex that binds to specific DNA sequences flanking the gene segments and introduces precise double-strand breaks. The breaks are then repaired by the non-homologous end-joining pathway, which ligates the segments together. The system is controlled genomic violence. The cell deliberately fractures its own DNA, creating breaks that would be lethal in almost any other context, and then relies on the repair machinery to stitch the pieces back together in a configuration that was not present in the genome it inherited. The process is highly regulated. The breaks occur only in developing lymphocytes, only at specific sites, only during a narrow developmental window. But the breaks are real, and the danger is real. If the repair fails, the lymphocyte dies. If the repair occurs incorrectly, if the wrong segments are joined, or if the breaks occur at unintended sites, the result can be chromosomal translocations, fusions between segments that should not be joined, instability that can lead to leukemia or lymphoma. Cancers of the immune system frequently carry translocations involving the antibody or receptor loci, scars of recombination gone wrong, but the risk is accepted. The immune system cannot function without diversity. A pathogen can mutate, can evolve, can present the host with molecules it has never encountered. The host's defense must be able to match that diversity, and the only way to generate it in real time is to rearrange the genome. The organism gambles with its DNA in order to survive. The discovery of how antibody diversity is generated earned Susumu Tonegawa the Nobel Prize in Physiology or Medicine in 1987. Tonegawa, working at the Basel Institute for Immunology in the mid-1970s, demonstrated that antibody genes are not inherited intact but are assembled during B-cell development through DNA rearrangement. The finding was revolutionary. It showed that the genome is not static, that gene sequences can differ between cells of the same organism, that the immune system achieves its flexibility by editing itself. Later work revealed that the RAG proteins evolved from a transposon, a mobile genetic element that once moved through genomes by cutting and pasting itself into new locations. At some point in vertebrate evolution, a transposon was domesticated, its cutting machinery repurposed for immune diversity. The system is ancient, conserved across all jawed vertebrates, fish, amphibians, reptiles, birds, mammals, and it is still at its core a controlled act of genomic instability. The paradox deepens when you consider where this is happening. The bone marrow produces roughly 500 billion new blood cells every day. A large fraction of those are lymphocytes, each one undergoing VDJ recombination during its development. 500 billion cells, each deliberately breaking its own DNA. The immune system is a production line of controlled damage, running continuously, and the repair machinery is the only thing standing between function and catastrophe. The same tension appears elsewhere. The intestinal epithelium replaces itself every 3 to 5 days. The lining of the gut is one of the most rapidly renewing tissues in the body, driven by stem cells at the base of small pits called crypts. Each crypt contains roughly 250 cells. A fraction of those cells divide twice a day. Divide, replicate the genome, divide again. The pace is relentless, the replication machinery running at capacity, and the margin for error is thin. In a developing embryo, the demands are even greater. A fertilized egg becomes a human being in 9 months, a process requiring trillions of cell divisions, each one a copying event, each one an opportunity for error. The genome is copied and copied and copied, and the fidelity must remain high, because errors in early embryogenesis are inherited by vast lineages of descendant cells. A mutation in a stem cell in week 3 of development may appear in billions of cells at birth. The genome is not a passive archive. It is an operational system, and the operations are expensive. Replication is expensive. Repair is expensive. And in the cells and tissues where the demands are highest, bone marrow, intestine, immune system, embryo, the machinery runs at the edge of its capacity. The fact that it works at all is extraordinary. 2 meters of DNA in every cell. 37 trillion cells in the body. If you laid all of it end to end, the total length would stretch roughly 67 billion kilometers, enough to reach the sun and back more than 200 times. All of it copied during development. All of it maintained across a lifetime. All of it damaged daily and repaired daily. The genome surviving not because it is chemically stable, but because the repair machinery is relentless. But there is a limit. At the ends of every chromosome there is a region that cannot be fully replicated. The problem is geometric. DNA polymerase synthesizes DNA in one direction only, from the 5' to the 3' end. On the leading strand, synthesis is continuous. On the lagging strand, it proceeds in short fragments, each initiated by a RNA primer. When the polymerase reaches the very end of the chromosome, there is no place to bind the final primer. The extreme tip cannot be copied. A small segment of DNA is lost with each round of replication. This is called the N-replication problem, and it is an unavoidable consequence of linear chromosomes and unidirectional synthesis. The loss is small, 50 to 200 base pairs per division, depending on the species and the cell type, but it is cumulative. Over many divisions, the chromosome shortens. To protect the coding regions of the genome, the ends of chromosomes are capped with repetitive sequences called telomeres. In humans, the telomere sequence is TTAG, repeated thousands of times. The telomeres are non-coding. They carry no genes. Their function is to absorb the loss. Each division shortens the telomere, not the chromosome's functional content. The telomere is a buffer, and the cell can divide as long as the buffer remains, but the buffer is finite. In normal human cells grown in culture, division continues for roughly 50 to 70 rounds before stopping. The number is called the Hayflick limit, named for Leonard Hayflick, who demonstrated in the early 1960s that human fibroblasts do not divide indefinitely, but enter a state of permanent arrest after a fixed number of divisions. The cells remain alive, metabolically active, but they no longer replicate. The state is called senescence. The cause, it was later shown, is telomere shortening. When the telomeres reach a critical length, the cell interprets the shortened chromosome ends as double-strand breaks. The damage response activates. Replication halts. The cell exits the cycle. This is not a defect, it is a safeguard. Senescence prevents cells with accumulated damage from continuing to divide, reducing the risk that mutations will be passed on. It is a clock, built into the genome structure, counting down with every division. But some cells need to divide indefinitely. Germ cells, which give rise to eggs and sperm, must transmit the genome to the next generation without shortening it. Embryonic stem cells, which generate every tissue in the developing organism, divide hundreds of times. These cells do not senesce, and their telomeres do not shorten, because they express an enzyme that adds new telomeric repeats to the chromosome ends. The enzyme is called telomerase. Telomerase is a ribonucleoprotein, a complex of protein and RNA. The RNA serves as a template, carrying the sequence TTAG, and the protein synthesizes new DNA using that template, extending the telomere outward and counteracting the end replication problem. The enzyme was discovered in the late 1980s by Elizabeth Blackburn and Carol Greider, working at the University of California, Berkeley, in experiments using the single-celled organism Tetrahymena. The discovery grew out of earlier work by Blackburn and Jack Szostak, who had shown in 1982 that telomeric DNA sequences from Tetrahymena could protect linear plasmids in yeast, suggesting the sequences had a conserved protective function. Greider and Blackburn identified the enzyme in 1984, demonstrating that cell extracts from Tetrahymena contained an activity that added telomeric repeats to DNA primers in vitro. They named it telomerase and showed that it contained both RNA and protein components. Later work confirmed that the enzyme is present in human germ cells and stem cells, active during development and largely silent in adult somatic tissues. The discovery explained how germ cells maintain telomere length across generations and how stem cells sustain long-term proliferation. It also raised a question. If most adult cells lack telomerase, how do cancer cells, which divide indefinitely, avoid senescence? The answer, it turned out, is that most cancer cells reactivate telomerase. Studies in the 1990s showed that telomerase activity is present in the vast majority of human tumours. Estimates range from 85 to 90%, but absent in most normal tissues. The reactivation is one of the key steps in the transformation of a normal cell into a cancer cell. Without it, the telomeres shorten, senescence is triggered, and the cell stops dividing. With it, the cell bypasses the Hayflick limit, its telomeres maintained, its capacity for division unlimited. Telomerase does not cause cancer, it enables it. The initial mutations that drive uncontrolled growth occur in other genes, genes that regulate the cell cycle, genes that suppress apoptosis, genes that promote angiogenesis. But without telomerase, those mutations can only take the cell so far. The tumor grows, divides, exhausts its telomeres, and senesces. Reactivation of telomerase removes that barrier. The enzyme became a therapeutic target. If telomerase is required for most cancers, but absent in most normal tissues, inhibiting it should selectively kill tumor cells while sparing normal ones. Drugs targeting telomerase have been developed, and some have entered clinical trials, though the results have been mixed. The challenge is that telomerase inhibition does not kill cells immediately. It shortens telomeres, but the effect takes time, dozens of divisions, before senescence is triggered. Rapidly dividing cancers may be slowed, but slowly dividing tumours may not respond within a clinically relevant time frame. Still, the principle is sound. The cell's own repair and maintenance machinery can become a vulnerability, and understanding the machinery opens the possibility of intervention. Blackburn, Grieder, and Szostak were awarded the Nobel Prize in Physiology or Medicine in 2009 for their work on telomeres and telomerase. The citation noted that their discoveries had illuminated how chromosomes are protected and how cells age, and had opened new avenues for understanding cancer and age-related disease. But the discoveries also deepened the central tension. Telomerase can extend lifespan, delay senescence, sustain the regenerative capacity of tissues. In experimental organisms, activation of telomerase has been shown to extend health span, delay the onset of age-related pathologies, maintain tissue function longer. The same enzyme, reactivated in the wrong context, sustains cancer. The genome is protected by layers of repair, by checkpoints and safeguards, by machinery that can detect and correct thousands of lesions every day. But those same systems, when altered, when lost, when reactivated inappropriately, can drive the diseases they were meant to prevent. Repair is not an unalloyed good. Unlimited repair, unlimited division, unlimited maintenance, these are not automatically blessings. They are tools, and the cell's fate depends on how and when they are used. The double helix is copied with an error rate of roughly 1 mistake per billion bases. It is damaged tens of thousands of times per day. It is deliberately cut during immune development. Its ends shorten with every division unless an enzyme intervenes. And across all of this, across 37 trillion cells, across a lifetime of 80 or 90 years, the genome remains mostly intact, mostly functional, the instructions still readable. The fidelity is not perfect, the system is not infallible, but it is sufficient, and sufficiency, in biology, is often enough. The question then is what happens when sufficiency fails? Not gradually, not through the slow accumulation of wear that marks aging, the greying hair, the stiffening joints, the cells that divide more slowly or not at all, but catastrophically, when the layers of defense, the proofreading, the mismatch repair, the base excision pathways, the checkpoints that halt division when damage is detected, when all of those layers are breached not by a single failure but by a cascade of them, and a cell that should have stopped dividing does not stop. This is the logic of cancer, and it begins with a single cell. Picture that cell, not as an abstraction, not as a diagram in a textbook, but as a physical entity 5 to 10 micrometers across, invisible to the naked eye, one among trillions. It contains 2 meters of DNA wound into 23 pairs of chromosomes, housed in a nucleus roughly 1/10 the width of a human hair. It is bathed in fluid, surrounded by other cells, connected by chemical signals, part of a tissue, part of an organ, part of a body. It has divided dozens of times already. It will divide dozens more if it is allowed to. But something has changed. A mutation, perhaps inherited, perhaps acquired through a copying error during replication, perhaps introduced by a chemical exposure or a burst of ultraviolet light. A single base altered, an adenine where a guanine should be, or a thymine dimer left unrepaired, or a deletion of several bases that shifts the reading frame. The mutation lands in a gene that regulates cell division, a gene whose normal function is to suppress uncontrolled growth. The protein it encodes is no longer made, or is made incorrectly, or no longer binds to the DNA sites it once regulated. The cell does not notice immediately. The mutation is recessive. One functional copy of the gene remains. The cell divides. The mutation is copied into both daughter cells. Years pass. Another mutation occurs, this time in the second copy of the same gene. Now both are non-functional. The brake is released. The cell begins to divide more frequently. Not wildly, not yet. But the signals that once told it to wait, to pause, to check for damage before proceeding, are no longer heeded. A third mutation occurs, this time in a gene that promotes division when activated. The mutation locks the gene in an active state. The cell divides again. A fourth mutation disables a gene involved in apoptosis, the programmed cell death that normally eliminates damaged cells. The cell, which should have been marked for destruction, survives. A fifth mutation affects a repair pathway, increasing the rate at which new mutations accumulate. The process accelerates, and still, for a time, the cell is held in check. Because there is one final safeguard, one final decision maker that integrates the signals from all the others and determines whether the cell will live or die. A protein that has been called, without exaggeration, the guardian of the genome. It is called P53. The protein was discovered in 1979, independently by several groups of researchers working on cancer-causing viruses. David Lane, working in London, was studying a virus called SV40, which could transform normal cells into cancer cells. He noticed that infected cells contained a protein that bound tightly to a viral protein. Arnold Levine, working at Princeton, identified the same protein in cells transformed by the virus. Lloyd Old and Pierre May, working in New York and France respectively, made similar observations. The protein had a molecular weight of 53 kilodaltons, and so it was named p53. At first, researchers believed p53 was an oncogene, a gene that promotes cancer. The protein was overexpressed in tumor cells, and when introduced into normal cells, it seemed to drive transformation. But the conclusion was based on a misunderstanding. The p53 found in tumors was mutated, non-functional. It was not driving cancer. It was a broken remnant of a protein that should have been preventing it. By the mid-1980s, the picture had reversed. Wild-type p53, the normal version, was shown to suppress tumor formation. It was not an oncogene. It was a tumor suppressor and one of the most important ever discovered. The gene that encodes p53, called TP53, is located on chromosome 17. It is mutated in more than half of all human cancers. In some cancer types, ovarian, colorectal, esophageal, the mutation rate is higher, approaching 70 or 80%. In others, even when TP53 itself is intact, the pathways that regulate p53 are disrupted. The protein is degraded too quickly, or never activated, or sequestered in the cytoplasm where it cannot function. The result is the same: the cell loses its guardian. The role of p53 is to sense damage and decide what happens next. The protein is normally present at low levels, constantly produced and constantly degraded, its half-life measured in minutes. But when the cell experiences stress, DNA damage, hypoxia, oncogene activation, ribonucleotide depletion, p53 is stabilized. It accumulates in the nucleus. It binds to DNA at specific regulatory sequences and activates or represses the expression of hundreds of genes. The genes it controls determine the cell's fate. If the damage is mild, p53 halts the cell cycle. It activates genes that produce proteins like p21, which inhibits the enzymes that drive progression from one phase of the cycle to the next. The cell pauses in G1, before DNA replication begins, or in G2, before mitosis. The pause gives the repair machinery time to work. Base excision repair removes oxidized bases. Nucleotide excision repair cuts out thymine dimers. Mismatch repair scans for replication errors. If the damage is repaired, p53 levels drop. The cycle resumes, the cell divides. The event leaves no trace. But if the damage is severe, if the repair machinery cannot fix it, or if the lesions are too numerous, p53 makes a different decision. It activates genes involved in apoptosis. Proteins like BAX and PUMA are produced. They migrate to the mitochondria, disrupt the outer membrane, and release cytochrome C into the cytoplasm. The release triggers a cascade of enzymes called caspases, which systematically dismantle the cell. The chromatin condenses, the membrane blisters, the cell fragments into membrane-bound vesicles that are quietly consumed by neighboring cells, or by immune cells passing through the tissue. The cell dies without inflammation, without releasing its contents, without disturbing the tissue around it. The body disposes of billions of cells this way every day. Most go unnoticed. Alternatively, p53 can push the cell into senescence, a state of permanent growth arrest. The cell remains metabolically active but never divides again. It secretes factors that alter the tissue environment, sometimes promoting repair, sometimes recruiting immune cells to clear away damaged neighbors. Senescent cells accumulate with age. Their presence is thought to contribute to inflammation and tissue dysfunction, though the full picture remains unclear. This is the trial. The cell's genome is interrogated, the damage assessed, the verdict delivered: repair, arrest, senescence, or death. p53 is not the only protein involved. There are upstream sensors, downstream effectors, feedback loops, redundancies, but it is the central node, the place where the information converges and the decision is made. It is in effect the judge. Now imagine what happens when the judge is removed. The cell sustains damage, a double-strand break perhaps, or a replication error that slips past the proofreading and mismatch repair. Normally, p53 would stabilize, halt the cycle, give the repair machinery time. but the TP53 gene is mutated. The protein is not made, or is made in a form that cannot bind DNA, or cannot activate its target genes. The cell does not pause, it proceeds into S phase. The damaged DNA is copied, the error is doubled, one copy in each daughter cell. The daughters inherit not only the original mutation in TP53, but also the new mutation introduced because the repair checkpoint was bypassed. The daughters divide, the process repeats. Each generation accumulates more mutations. Some are neutral, affecting regions of the genome with no functional consequence. Others land in genes that regulate division, apoptosis, DNA repair, angiogenesis, immune recognition. The mutations that confer a growth advantage, faster division, resistance to apoptosis, independence from external growth signals, are selected for. The cells that carry them out compete their neighbours. The population expands. 3 to 10 driver mutations across several key pathways are often sufficient to transform a normal cell into one capable of founding a tumour. The number is small, and that is part of what makes cancer so difficult. The genome is 3 billion bases long, but only a tiny fraction of it, perhaps 1 to 2%, codes for proteins, and only a subset of those proteins are involved in the regulation of growth and survival. A single cell divides perhaps 50 times in a human lifespan. With an error rate of 1 per billion bases per division, and with tens of thousands of additional lesions per day, the chance that any given cell will acquire all the necessary mutations is low. But the body contains 37 trillion cells, and each is a separate lottery ticket. Run the lottery long enough, and eventually one cell wins, or loses, depending on the perspective. That one cell divides, its descendants divide. Within a few generations, the population numbers in the thousands. Within a few more, millions. The cells begin to crowd the tissue, displacing normal cells, disrupting architecture. They secrete factors that stimulate the growth of new blood vessels, diverting nutrients and oxygen to the expanding mass. They evade immune surveillance, either by downregulating surface markers that would flag them as abnormal, or by secreting signals that suppress immune activity. Some acquire the ability to invade surrounding tissues, breaking through basement membranes, migrating into the bloodstream or lymphatic system, seeding new growths in distant organs. By the time the tumour is detectable, by imaging, by biopsy, by the symptoms it causes, it may contain billions of cells, all descended from that single ancestor, all carrying the mutations that allowed it to escape. The statistics are sobering. Mutations in TP53 or its regulatory pathways are found in more than half of all cancers. Individuals born with one mutated copy of TP53, a condition called Li-Fraumeni syndrome, named for Frederick Lee and Joseph Fraumeni, who described it in 1969, face a 50% risk of developing cancer by age 30 and a lifetime risk approaching 90%. The syndrome is rare, affecting perhaps 1 in 5,000 to 1 in 20,000 individuals, but it lays bare what happens when even one layer of defense is compromised from birth. Every cell in the body carries the mutation. Every cell is one step closer to malignancy. The cancers that arise are diverse: sarcomas, brain tumours, breast cancers, leukemias, but they share a common origin: the absence of a functional guardian. The cascade is not inevitable. Most cells with damaged TP53 do not become cancer cells. Additional mutations are required in other genes, in the right combination, in a permissive tissue environment. The immune system clears many abnormal cells before they can establish a foothold. The body's architecture, the physical constraints of tissue, the availability of blood supply all impose barriers. But the risk is real, and it grows with time. The longer a cell lives, the more divisions it undergoes, the more opportunities there are for the necessary mutations to accumulate. This is why cancer incidence rises steeply with age. The genome does not degrade uniformly. It accumulates insults, one after another, until the burden overwhelms the defenses. And yet, most people do not develop cancer. Most cells, across a lifetime of 80 or 90 years, remain normal. The machinery works. The proofreading catches most errors. The mismatch repair scans the newly synthesized strands and corrects the errors the proofreading missed. The base excision repair removes damaged bases. The nucleotide excision repair excises bulky lesions. The double-strand break repair pathways, homologous recombination and non-homologous end joining, restore severed chromosomes. The checkpoints halt division when damage is detected. And P53, when it is functional, makes the final call: repair, arrest, or die. The system is layered, redundant, robust. It is not perfect. It was never designed to be perfect. But it is sufficient to protect the genome across trillions of cells and billions of divisions to hold the error rate low enough that most cells never acquire the mutations necessary for malignancy, to ensure that the instructions written in the double helix remain readable, mostly intact, through the decades of a human life. The revelation is not that the system works flawlessly, it is that it works at all. That 2 meters of DNA, subjected to constant chemical assault, copied 50 times or more, exposed to radiation, oxidation, hydrolysis, mechanical stress, can be maintained with an error rate measured in parts per billion. That the machinery required to achieve that fidelity, the polymerases, the ligases, the helicases, the exonucleases, the damage sensors, the checkpoint proteins, the hundreds of repair enzymes working in shifts around the clock, exists in every cell, continuously active, continuously scanning, continuously correcting. That the genome is not a static archive but a living document, constantly revised, constantly under threat, and constantly defended. The defense is not passive, it is active, dynamic, responsive, and it is the product of the same evolutionary process that produced the genome itself. The cells that could not maintain their DNA did not survive. The lineages that could not repair damage, that could not suppress tumors, that could not regulate division, were eliminated. The machinery we observe now is what remains after billions of years of selection, the minimal set of tools required to keep a genome intact long enough to reproduce, to pass the instructions to the next generation, to sustain the lineage. But sufficiency, in biology, has limits. The system is optimized for the environments and lifespans in which it evolved. Humans now live longer than their ancestors, exposed to chemical and radiological hazards that did not exist a century ago, eating diets, and engaging in behaviors that alter the burden of DNA damage. The machinery still works, but it is strained. The error rate is low, but it is not zero. And across 37 trillion cells, even a low rate compounded over decades produces a non-negligible risk. The risk can be managed. Screening programs detect cancers early, when they are small, localized, treatable. Genetic testing identifies individuals at high risk, allowing for increased surveillance or preventive measures. Therapies targeting the molecular machinery of cancer, inhibitors of telomerase, of DNA repair, of checkpoint proteins, exploit the vulnerabilities introduced by the mutations the cancer cells carry. The principle is the same as the one underlying PARP inhibitors. Find a pathway the cancer cell depends on, block it, and let the cell's own instability destroy it. But intervention is not without cost. Chemotherapy and radiation, the mainstays of cancer treatment for decades, work by damaging DNA, by overwhelming the repair machinery, by forcing cells into apoptosis. They are effective because cancer cells, dividing rapidly, are more vulnerable to DNA damage than most normal cells. But normal cells are damaged too: bone marrow, gut lining, hair follicles, any tissue with a high rate of division. The side effects are the cost of the treatment, the collateral damage of a strategy that targets the genome itself. Newer therapies are more selective. Targeted drugs inhibit specific proteins overexpressed or mutated in cancer cells. Immunotherapies train the immune system to recognize and kill tumor cells. CAR T-cell therapy engineers a patient's own T-cells to target cancer antigens. These approaches are more precise, and in some cases extraordinarily effective, but they too carry risks, off-target effects, immune overactivation, resistance, as cancer cells mutate further and evade the therapy. The genome's mutability, the very feature that allows evolution, also allows cancer to adapt. The tension runs through every intervention. The tools that maintain the genome can become vulnerabilities. The tools that kill cancer cells can damage normal ones. Precision comes at the cost of complexity. Complexity introduces the possibility of failure, and still, the work continues. Because the alternative, doing nothing, accepting the risk, allowing the machinery to fail without response, is not acceptable. The genome is the foundation of every cell, every tissue, every organ, every function that sustains a human life. When it is protected, the body thrives. When it is damaged, the body suffers. And so the effort to understand the machinery, to measure its fidelity, to identify its failure modes, to design interventions that shore up its defenses or exploit its vulnerabilities continues. Return now to the body as a whole. 37 trillion cells, each containing 2 meters of DNA, each copying that DNA dozens of times, each repairing tens of thousands of lesions every day. If the DNA from every cell were unspooled and connected end to end, the strand would stretch roughly 74 billion kilometers, enough to travel from Earth to the Sun and back not once, not 10 times, but more than 200 times. The scale is staggering, and yet the system works. The instructions are copied, the damage is repaired, the cells divide, function, die, and are replaced. The work is invisible. You do not feel the polymerase moving along the replication fork. You do not sense the mismatch repair protein scanning the newly synthesized strand. You do not notice the p53 accumulating in the nucleus of a cell that has just sustained a double-strand break, halting division, giving the repair machinery time. But the work is constant and it is essential. Without it, the genome would degrade within days. The cells would die, the tissues would fail, the body would collapse. The custodians of the code are not conscious. They are proteins, enzymes, molecular machines assembled from amino acids, folded into precise three-dimensional shapes, operating according to the laws of chemistry and physics. They do not choose to repair DNA. They do it because that is what they do, what they have been shaped by evolution to do, what their structure allows them to do. And yet, the cumulative effect of their work is the preservation of information across time, the maintenance of a message written 4 billion years ago and continuously revised, continuously passed down, continuously read. The custodians include the discoveries that revealed them. Friedrich Miescher, isolating nuclein from the nuclei of white blood cells in a Castle laboratory in Tübingen, with no knowledge of what he had found. Rosalind Franklin, capturing the diffraction pattern of DNA with X-ray crystallography, an image so clear it pointed directly to the double helix. Thomas Lindahl, measuring the instability of DNA and discovering that it is not a permanent archive, but a structure under constant repair. Elizabeth Blackburn, identifying the telomeres at the ends of chromosomes and the enzyme that maintains them, revealing both the mechanism of aging and a pathway exploited by cancer. Each discovery built on the ones before it. Each answered a question and raised new ones. The structure of DNA suggested a mechanism for replication, but not the machinery that performed it. The discovery of repair enzymes explained how damage was removed, but not how the cell decided when to repair and when to die. The identification of telomerase explained how cells could divide indefinitely, but also how that capacity could be hijacked. The genome was no longer a static blueprint. It was a dynamic system, constantly under threat, constantly defended, constantly balanced between stability and change. The tools to interrogate that system have advanced in parallel. Genome sequencing, once a laborious process requiring years and millions of dollars to sequence a single human genome, can now be done in hours for less than $1,000. The sequence is no longer a mystery. It is data, searchable, comparable, and notable. Mutations can be catalogued, pathogenic variants identified, cancer genomes sequenced to reveal the mutations that drive them and the vulnerabilities that can be targeted. CRISPR, the genome editing tool adapted from a bacterial immune system, allows DNA to be cut and altered at specific sites with a precision that was unimaginable a generation ago. The technology is being used to correct disease-causing mutations in cells grown in the lab and in some cases in patients. Clinical trials are underway for sickle cell disease, for beta thalassemia, for inherited blindness, for cancers resistant to other treatments. The promise is real. The risks are real too. Off-target cuts, unintended mutations, mosaicism where only some cells are edited, all are possible. The consequences may not be apparent for years. Anti-CRISPR proteins discovered in bacteriophages are being developed as safety switches, a way to halt the editing process if something goes wrong. The field is still young. The rules are still being written. Newborn screening programs test for genetic disorders, many of them caused by mutations in DNA repair genes, in metabolic enzymes, in structural proteins. Early detection allows for early intervention, sometimes preventing symptoms entirely. Cancer genomics tailors treatment to the specific mutations present in a patient's tumor, selecting drugs likely to be effective and sparing patients from therapies unlikely to help. DNA repair-based therapies exploit the dependence of cancer cells on alternative pathways, creating synthetic lethality, selectively killing the cells that have lost their guardian. All of this rests on the understanding of the machinery, the custodians, the molecular systems that copy, maintain, repair, and defend the genome. The understanding is incomplete. There are genes whose functions are unknown, repair pathways whose mechanisms are not fully characterized, interactions between pathways that remain unclear. But the picture is far more complete than it was 50 years ago, or 20 or 5, and yet the genome remains, in some fundamental way, beyond full control. It can be read, measured, edited, but not commanded. It is the product of billions of years of evolution, shaped by selection pressures no longer operative, carrying within it the history of every ancestor, every mutation that survived, every pathway that proved useful. It is robust and fragile, stable and mutable, exquisitely precise and prone to error. It is the foundation of life, and it is constantly on the edge of falling apart. Every breath depends on it. Every heartbeat, every thought, every movement, every immune response, every wound that heals, every cell that divides to replace the ones that have died. The instructions are there. In the nucleus of every cell, 2 meters of DNA coiled into a space 1/100th the width of a dot on a page, read out in sequences of RNA, translated into proteins, assembled into the structures that perform the work of being alive. The custodians work without rest.